हेलो एंड वेलकम टू बॉटनी क्लासरूम। इस चैनल पर आपको मिलेंगे बॉटनी ऑनर्स माइनर एंड एमडीसी एंड ऑल्सो जूलॉजी ऑनर्स माइनर एंड एमडीसी के बहुत सारे सॉल्व पीवाईक्यू एंड सजेशंस क्वेश्चंस। आज हम इस वीडियो में पढ़ेंगे सेल बायोलॉजी के इलेक्ट्रोस्कोप चैप्टर के थोड़े 5 मार्क्स के लॉन्ग क्वेश्चन आंसर्स जो आपके एग्ज़ाम में आने के इसके चांसेस बहुत ज़्यादा है। Question like difference between SEM and TEM and what are the disadvantages and advantages of electron microscope। ये दो क्वेश्चंस थोड़े ज़्यादा इम्पॉर्टेंट है क्योंकि बहुत बार पूछा जा चुका है आपके PYQs में, तो उसको थोड़ा ध्यान से पढ़ें। वीडियो को स्टार्ट करते हैं हमारे क्वेश्चन नंबर 1 से। क्वेश्चन नंबर 1, व्हाट इज द वर्किंग प्रिंसिपल ऑफ इलेक्ट्रॉन माइक्रोस्कोप। आंसर, electron microscopes use signals arising from the interaction of an electron beam with the sample to obtain information about structure, morphology, and composition. Point number 1, the electron gun generates electrons. Number 2, two sets of condenser lenses focuses the electron beam on the specimen then into a thin light beam. Point number 3, to move electron down the column, an accelerating voltage mostly between 10 kilovolt to 1000 kilovolt is applied between the tungsten filament and anode. Point number 4, the specimen to be examined is made extremely thin, at least 200 times thinner than those used in the optical microscope. Ultra thin sections of 20 to 100 nanometer are cut, which is already placed on the specimen holder. Point number 5, the electronic beam passes through the specimen and electrons are scattered depending upon the thickness or refractive index of different parts of the specimen. Point number 6, the denser region of the specimen scatter more electron and therefore appear darker in the image since fewer electrons strike that area of the screen. In contrast, transparent regions are brighter. Point number 7, the electron beam coming out of the specimen passes to the objective lens, which has high power and forms the intermediate magnified image. Point number 8, the ocular lens then produces the final further magnified image. Question number 2, what is the difference between SEM and TEM? Properties SEM and TEM, ऐसे 3 रोज बना लेना है। Point number 1, प्रॉपर्टीज का SEM का होगा, it is based on scattered electrons that are emitted from the surface of a specimen. TEM, it is based on transmitted electrons. Number 2, sample preparation. Sample can be of any thickness and is coated with a thin layer of heavy metal such as gold or palladium and mounted on an aluminum slab. TEM, laborious sample preparation is required. The sample has to be cut into thin sections so as to allow electrons to pass through it and are supported on TEM grids. Point number 3, resolution. The resolution is up to 20 nanometer. TEM has much higher resolution than SEM. It can resolve objects as close to 1 nanometer. Point number 4, magnification. The magnifying power of SEM is up to 1 lakh x. Magnifying power of TEM is up to 50 lakh x. Image formation. SEM provides a 3-dimensional image. Secondary or back-scattered electron are displayed on computer screen. TEM provides a 2-dimensional image. Transmitted electron hit a fluorescent screen, giving rise to a shadow image. The image can be studied directly by the operator of photograph with a camera. Point number 6, SEM is used to study the topography and atomic composition of specimens. TEM is used to study the interior of cells, the structure of protein, molecules, the organization of molecules in iris and cytoplasm. cytoskeleton, filaments, and the arrangement of protein molecules in cell membrane. Question number 3. Describe parts of electron microscope. Electron microscope is in the form of a tall vacuum column that is vertically mounted. It has the following components. Point number 1. Component number 1 is electron gun. The electron gun is a heated tungsten filament which generates electrons. Number 2. Electromagnetic lenses. The condenser lens focuses the electron beam on the specimen. A second condenser lens forms the electrons into a thin light beam. The electron beam coming out of the specimen passes down the second of magnetic coils called the objective lens, which has high power and forms the intermediate magnified image. Next point, the third set of magnetic lenses called projector ocular lens produce the final further magnified image. Each of these lenses acts as an image magnifier, all the while maintaining an incredible level of detail and resolution. Next, specimen holder. The specimen holder is an extremely thin film of carbon or colloidal held by a metal grid image viewing and recording system. Point number 1. The final image is projected on a fluorescent screen. Point number 2. Below the fluorescent screen is a camera for recording the image. Question number 4. How many lenses are used in electron microscope? There are 3 different lenses in electron microscope, such as the condenser lens, objective lens, and projector lens with different functions. Point number 1. The condenser lens focuses the electron beam on the specimen. A second condenser lens forms the electrons into a thin tight beam. Second point, the electron beam coming out of the specimen passes down the second of magnetic coils called the objective lens, which has high power and forms the intermediate magnified image. Third set of magnetic lenses called projector ocular lenses produce the final further magnified image. Point number 4. Each of these lenses act as an image magnifier, all the while maintaining an incredible level of detail and resolution. Number 5. What are the sources of electron in electron microscope? Answer. Point number 1. The electron gun is a heated tungsten filament which generates electron. Point number 2. 2 sets of condenser lenses focus the electron beam on the specimen, then into a thin tight beam. Point number 3. To move electrons down the column, an accelerating voltage, mostly between 100 kV to 1000 kV, is applied between the tungsten filament and anode. What is the role of anode in electron microscope? Answer. The anode receives the flow of electrons, electron beam emitted from the cathode, and guides the electron flow downwards through the hole or at its center. 7. What are the advantages and disadvantages of electron microscope? Answer. Advantages. Point number 1. Very high magnitude, incredibly high resolution. Material. Point number 3. Material rarely distorted by preparation. Point number 4. It is possible to investigate a greater depth of field. Point number 5. Diverse application. Now the disadvantages. Number 1. Live specimen cannot be observed. Point number 2. As the penetration power of the electron beam is very low, the object should be ultra thin. For this, the specimen is dried and cut into ultra thin sections before observation. Point number 3. As the EM works in a vacuum, the specimen should be completely expensive to build and maintain. Point number 5. Requiring researcher training. Point number 6. Image artifacts resulting from specimen preparation. Point number 7. This type of microscope is large, cumbersome, extremely sensitive to vibration and external magnetic field. ये वीडियो अच्छा लगा हो तो जल्दी से चैनल को सब्सक्राइब करें, लाइक शेयर एंड कमेंट भी करें कि आपको ये वीडियो कैसा लगा। थैंक यू।